Hello everyone. My name is Mohsin Qureshi. I am a PhD student in Department of Biomedical Science and Engineering, GIST, and my PhD supervisor name is Ihan Chang. Today I will present my research topic with the title of Measuring Blood Flow in the Mouse Brain During Stroke by Quantitative Laser Speckle Velocimetry based on image correlation. So here are the contents of the presentation. This presentation is divided into 4 parts. The introduction, second one is quantitative laser speckle imaging, and then we will validate it through solid diffuser and RBCs in microfluidic channel, and last is the in vivo application of this procedure. The human brain, even though being nearly 2% of the whole body mass, consume 20% of body's oxygen and nutrients supply, for example glucose. The microvasculature system and the cerebral blood flow play a crucial role in transporting oxygen and glucose in the brain. Measuring blood flow in cerebral microvessel can be potential biomarker for studying the normal and pathological condition of brain metabolism for both clinical and research settings. In the field of biomedical imaging, scientists and researchers focused on measuring and monitoring tools for spatiotemporal measurement of cerebral blood flow. Therefore, methods for measuring cerebral blood flow have divided under 3 categories. The first category is Doppler-based method, for example laser Doppler flowmetry and Doppler optical coherence tomography. The second method is red blood cells tracking measurements, for example intravital microscopy, and the third one is laser speckle contrast imaging. Even though each of these techniques is different in terms of instrumentation and analysis, however, each method was developed on the ground of dynamic light scattering. Recently, speckle-based cerebral blood flow imaging tools have gained popularity because it is simple and has high spatial resolution but semi-quantitative. All the particles in the biological sample have random motion. When a laser light interacts with the dynamic scattering medium like any biological medium, then dynamic speckle pattern produced over the time interval of t. These speckle patterns have the information of dynamic scatter in the sample. We report a new laser speckle based absolute blood flow measurement technique and adopt the spatiotemporal cross correlation technique. Then we validate the quantitative laser speckle imaging method both in translating diffuser and in vivo and in vitro sample of flowing RBCs in the microfluidic channel. Later, we demonstrate the quantitative laser speckle imaging in photothrombosis model, in which we show the map of velocity for the pre-stroke, during the stroke, and post-stroke condition. To the best of our knowledge, our technique is the first one using laser speckle that provides a velocity map, including both velocity magnitude and its direction for the in vivo study. The quantitative laser speckle imaging technique requires a minimum of 2 consecutive images, which is shown here as first and second frame. As this technique is based on spatiotemporal cross-correlation of 2 consecutive laser images with the time interval of delta t. We select a small square-shaped region, we call it as a probing window. Window on 2 consecutive images are on the same location. Taking the Fourier transform of probing window and multiply the first window with the conjugate of the second window, this gives the cross correlation map for that specific window. All these steps will be repeated for n realization. In this study we took n is equals to 4. We took the average of n cross correlation maps and find out the maximum peak displacement from the center of the map. And thus we can find the local speed by dividing the displacement by the frame rate of the camera. Afterward, the probing window moves one pixel and the procedure was repeated. In this way, the whole image were scanned by the probing window and get the correlation map. Experimental setup of quantitative laser speckle imaging is shown in this figure. Speckles were formed by green laser of wavelength 532. With the help of mirrors, laser beam was moved to the AOM, which was connected to function generator, and the first order diffraction beam passed through the iris. The modulated output of the AOM was expanded by a pair of convex lens and illuminate over the sample. The translating diffuser is a third level diffuser on a stage that moves from right to left side. The detection part consists of microscope objective lens. The captured light passes through an iris that increases the speckle size, a linear polarizer for improving the speckle contrast, and a tube lens of focal length 180 cm is used. Finally, the light was captured by the SCMOS camera. Figure A, B, and C shows the result of 3, 5, and 7 mm per second speed of the linear translator. The arrowhead shows the direction of glass diffuser movement, and the length of arrows shows the magnitude of velocity. Figure D shows the graph between the translation speed of linear translator and estimated speed of quantitative laser speckle imaging. So, we conclude that the maximum velocity that we estimate from quantitative laser speckle imaging is up to 7 mm per second. Here the experimental setup is similar to the linear diffuser experiment, but the sample changed here. For the in vitro validation, we used the RBC flow in the microfluidic channel. The length of the channel was 20 mm, width 1 mm, and height 45 mm. The inlet diameter was 1 mm and outlet diameter was 2 mm. For the steady flow, we used a syringe pump with the control flow rate. RBC flow from left to right side. Figure A, B, and C shows the result for 1.5, 2.2, and 3.7 mm per second particle speed calculated by the syringe pump flow rate. The flow direction shows the flow direction and the length shows the magnitude of velocity. Figure D shows the graph between the preset speed and the estimated speed from the quantitative laser speckle imaging. Therefore, we can say that with the fluid phantom, we validate the technique of quantitative laser speckle imaging. To show the application of quantitative laser speckle imaging, we experiment with the photothrombosis stroke model. To do the photothrombosis model, we perform the cranial window over the mouse skull. In this model, we took speckle images at pre-stroke, during a stroke, and post-stroke condition. Before the photothrombosis, speckle images were recorded. Then rose bengal was injected by bolus injection, and from another laser, green light was shined for 2 minutes. In the middle of 2 minutes, that is, after 1 minute, speckle images were captured during the photothrombosis procedure. After the photothrombosis procedure, we took speckle images after 4 minutes. The velocity, direction, and magnitude maps for pre-stroke are shown in figure A and B. For during stroke condition, it is shown in C and D. And for the post-stroke condition, the map and the velocity magnitude are shown in figure E and F. The refreshing rate of each map is 40 maps per second. Furthermore, it shows clearly that the flow reduces from pre-stroke to post-stroke condition. Conclusion. So, we create a novel quantitative laser speckle imaging technique used for making velocity maps for in vivo cases. We validate our technique by translating diffuser and in vitro samples. The maximum velocity which can be measured by quantitative laser speckle imaging is up to 7 millimeter per second. We also demonstrate velocity map in a stroke model. Thank you. If you have any question, feel free to ask.